Okay, thank you very much. Thank you for all the organizers for having me here, the opportunity to tell you a little bit about our work. I added another part of the title of my talk, so I have also to apologize because there will be some chromatin in my talk, but not much. So you can think about it as a break from all the chromatin that you learned about. Okay, so we are a single molecule biophysics lab, and we mainly use optical tweezers to study interactions between proteins and DNA, including chromatin, in the context of transcription. So, as you all know, binding of transcription factors to their sequence-specific binding sites and regulatory regions is a crucial event in the initiation of transcription. And it is highly regulated by many regulatory cues. For example, there are many binding sites for the same transcription factor on a promoter. There are different transcription factors that bind to the same promoter, so they can either cooperate or interfere with each other. The DNA is heavily modified, for example, by methylation of CpG dinucleotides. And of course, there are nucleosomes. And all these different regulatory marks contribute to regulating the accessibility of these transcription factors to the DNA. Now, many of the same regulatory cues that you see at regulatory regions are also present, and I forgot to say histone variants and modifications of the histones. But many of these marks are also present in the gene body. As you all know, gene bodies are also packaged into chromatin. There are also specific histone variants or modifications of the histone proteins. And transcription factors that have a very short binding site between 5 and 15 base pairs, they can appear just randomly in the bodies of genes. And some of them are covered by nucleosomes, but many are not. And if they are there, proteins are in the solution, they can also bind. So what this all means is that even though we always look with particular attention at the promoters, at the regulatory regions, this whole marks will also have an effect during elongation of transcription, because the RNA polymerase will have to cope with nucleosomes on the DNA, right? And there has been a lot of work on how exactly the polymerase copes with that, including structural work and even single molecule work looking at that. But polymerase also has to cope with small proteins, transcription factors that just perhaps randomly bind to the DNA. And this is a less well-studied question. How does the polymerase copes with these obstacles? So what I'm going to do today is actually, first I will tell you about some of the tools, some of the assays we have developed in my lab in the last few years. And there will be a little bit of chromatin there. And then after telling you about all these different tools, I will tell you how we approach this question of the encounter between the polymerase and the transcription factor using the tools I just mentioned. Okay, so this is the idea. So I divided them in three different aspects. So the first aspect will be the thermodynamics of a protein DNA complex. So the way that we do our experiments, imagine that you have a sample. In this case, it's just a piece of DNA. We close this DNA on one side, attach to each one of the strands of the double-stranded DNA another DNA molecule that has some chemical moiety on the end. So we can tether this whole complex between two bits, each of them trapped in an optical trap. We can now start to apply a force on this whole complex. So at the beginning, this is force versus extension. At the beginning, you mainly see stretching of the handles, right? So this is not interesting right now. But if you reach a high enough force, you start to destabilize the hydrogen bonds that keep together the two strands. So you see this type of sawtooth pattern. Each one of these drops in force and grow in extension is the opening of a few base pairs of DNA. By the time you reach the loop, you can continue stretching, as you can see here, and you can go back and forth, and it's completely reproducible and completely reversible also. So since we understand very well this elasticity of DNA, we can convert this curve, force versus extension, into a curve of force versus the number of base pairs unzipped. So anything that is only stretching will be a vertical line, right? And you see all these different events. Okay, why is this useful? Because what happens if you have now a protein in the solution that can bind the DNA someplace? What happens is that when you try to open the DNA, you bump into the protein. You need to increase the force to a much higher value until you can break the interactions of the protein with the DNA. The protein goes back into the solution, and you can continue. You can continue unzipping the DNA. So from this type of experiment, you can extract the position of a bound protein. You can extract how much force you need to break it. And if you repeat this experiment many times and you let the system equilibrate in between the unzipping cycles, you can extract the binding probability, which actually tells you something directly about the affinity of the protein for the DNA. So an example of the things that we can do with this, a few years ago, we looked at this transcription factor, EGR1. It's a zinc finger transcription factor, 3 zinc fingers. Each one makes contacts with the triplets of base pairs. So we look at our favorite gene. It's called LHB. And in the promoter of LHB, there are 3 binding sites for EGR1. But each one of them is different than the others. This is the consensus binding site for EGR1. As you can see, each one of these binding sites is different from the consensus. If you look across evolution, you see that these deviations from the consensus are conserved. So they are important for some reason to regulate this gene using different binding sites, each one of them with some different properties. So we use our assay to look at the promoter of LHB. And as you can see here, when you introduce EGR1, you see 3 peaks. So there are 3 binding sites for EGR1. You can, as I said, you can extract the breaking force, the binding probability, and from that, we could conclude that both the core sequence to which the protein binds and also the flanking sequences modulate both the affinity and the structure of the DNA. And if you want, I can say after that why I say structure. I don't want to dwell too much on that. Okay. Now, this is nice, but this experiment was done at thermodynamic equilibrium. For example, if you increase the time between cycles, what we call the incubation time, you increase it from 40 to 160 seconds, you measure exactly the same binding probability. So this is thermodynamic equilibrium. However, there are more and more papers that show that what really is important for the regulation of many genes is not the binding probability or the occupancy, it's the kinetics of binding and dissociation. For example, you can have 2 scenarios with 50% occupancy, one with fast interchange between binding and unbinding, and the other one slow, and they will have different regulatory properties. So we asked the question, can we use our methods to measure also the kinetics of binding? So we decided to do an assay in which when we unzip the DNA, we reach the position of the binding site. Instead of continue unzipping, we just stop there. And now we go completely passive. So in our experiment, the DNA is just held between these 2 harmonic traps. And just because thermal energy, the DNA can fluctuate, can do some breathing fluctuations of this region, right? So we can see this, you can see this as fluctuations. You zoom in, you see it's completely 2-state system, open and close. And this is about maybe 20 base pairs of DNA. So now if you add the protein, since the protein can bind the double-stranded form of the DNA, you see suppressions of the fluctuations every time a protein binds and fluctuations again when it dissociates. So you can measure hundreds and thousands of binding and dissociation events. You can create the whole probability distribution for the binding time, dissociation time. And if these are exponentially distributed, you can extract directly K on and K off. By the way, I would say that the most interesting cases, in my view, would be when this is not exponentially distributed, right? Because then our system can tell you something that it's much more powerful than other methods to measure the kinetics of binding and dissociation. And another point I want to measure about this method is that these are completely unlabeled proteins. We don't need to label them in any way. And they also bind to the DNA when the DNA is in a relaxed state because they bind to the hairpin, right? Okay. An example of what we can do with this, we look at what happens when we methylate the binding site of this protein. So the consensus binding site has 2 methylation sites. We saw that this doesn't affect at all the K on, but there's a huge effect on the K off for the protein. We repeated this experiment with the binding sites seen in the LHB gene. Each one of them has a different methylation pattern. And we saw, we'll do it quickly, what we saw is that the methylation effect can be both stabilizing or destabilizing depending on the position of the methylation site, whether it's on the right side or the left side. Okay, so we have thermodynamics, we have kinetics. What can we say about the structure and the structural dynamics? So you can, well, as you all know, I can skip this slide. We saw many versions of this. So what happens if instead of taking a small protein that can bind from the solution to the DNA, we have a nucleosome, right? So this is just DNA, and now it doesn't bind from the solution. It's a nucleosome that was reconstituted in advance on the DNA and then ligated to our construct. So when you unzip this, what you see is a much richer structure than a single protein. You see different breakings that represent breaking of different interactions between the histone proteins at the DNA, and you can characterize them all. So this idea, this method was pioneered by Michel Wang's lab, and we did all sorts of further advances on this. So, so for example, one example of what you can do with this structural approach is we look at this is the unzipping pattern of a nucleosome. You see the interactions with H2A and H2B, interactions with H3 and H4 at the dyad of the nucleosome. So what happens if you now add linker histones to the problem? So what we expected to see that we will see interactions of the linker histone with DNA here, the linker DNA region, and not to see any change here at the interaction of the core histones with the DNA. Actually, what we saw was exactly the opposite. We didn't see anything here, practically, and we saw a big stabilization of the histone core interactions with the DNA, just by the presence of the linker histone. You can take it further and take many of these curves and create this interaction map where I'm showing you something called the force-weighted dwell time, which is a proxy for the energy of interaction. In black, you see the nucleosome, and in red, you see the chromatosome. So what about the interactions of the linker histones with the linker DNA that we didn't see? So one of the reasons we didn't see them is that opening the 601 sequence at this region requires quite a high force because of its GC content. So we prepared another DNA sequence that conserves the 601 at the middle where the tetramer binds, but has an 80 rich sequences in the periphery. So when we did that, you can see this in black here. This would be here the first nucleosomal, the H2A, H2B interaction. So everything now is at the lower force. So using this construct and using another method, which means that instead of breaking the nucleosome just in one time all the way, we just unzip the DNA all the way to the first interaction of the core histones. We don't break them, we just go back and forth, back and forth, back and forth. So we can gather a lot of data. And as you can see here in the blue curve, we could identify specific interactions of the linker histones with the linker DNA, right? And one of the interesting things that we saw is that these interactions are very, very extended, right? This is about 150 base pairs from the dyad. So if you go to the other side too, that means that this chromatosome particle can have interactions in a region of 300 base pairs almost, which is much bigger than what you see in footprint experiments. And another interesting thing that I want to mention is that if you look at the binding probability of these interactions, you know, how often we see these interactions when we do many cycles, they are all smaller than 1, meaning that these interactions are dynamic. They can form and dissociate, form and dissociate. So they're not fixed interactions, but they represent also the dynamics of the conformation of the nucleosome. Another example of what we can do with this method, so a few years ago, we looked we used this idea of repetitive probing the nucleosome without breaking any of its interactions to measure the position of nucleosomes many times with time. So the same nucleosome, we just measure its position once and again, So we can measure, the diffusion of the nucleosome on the DNA, and we could show that the introduction of the variant H2AZ increases the diffusional properties of the nucleosome. This is shown here also with the mean square displacement. And we also showed that this increased diffusion actually facilitates binding of transcription factors. Okay. How am I going with the time? 10 more minutes. Okay, I have to rush. So now we want to use all these methods to study this problem. And what I'm going to say now is all unpublished. So the first thing that we said, okay, if we can measure the position of a protein by measuring the suppression of the fluctuations, perhaps we can also use this for RNA polymerase, right? And indeed, if you have an RNA polymerase on the DNA, this suppresses the fluctuations. So we did an experiment in which we started with a fluctuating DNA, and an RNA polymerase did this far away, maybe 20 base pairs or 30 base pairs. So we will see the fluctuations. And then at some point, we use a laminar flow cell to move this whole construct into a region that contains RNTPs, right? So this is what we see. You see fluctuations, and at some point, you see suppression, right? So by measuring the time it takes from the moment you expose the construct to the RNTPs to the moment you see a suppression of fluctuations, we can measure the passage time, how long it takes the polymerase to cross this region. Okay, and you can see that it also depends on the concentration of the nucleotides. Okay, so now we can make a construct in which in between the polymerase and the fluctuating region, there is a binding site for EGR1. When you do that, and now you repeat the experiment with EGR1 everywhere in the chamber, you can see that the passage time becomes much longer in the presence of EGR1. So even this small protein that binds only 9 base pairs of DNA is capable of delaying the polymerase for quite a long time. So this is just a typical trace, and this is the full distribution, right? You can see that this is very, very different. So one possibility is that the polymerase is completely passive. So it moves until it reaches the position of the binding of the transcription factor, and it has to wait until the transcription factor dissociates. So to check that, we can look, we take the passage time, we subtract the time it takes to passage without the EGR1, and we call this the interaction time. So this is the time actually spent interacting with the EGR1. So we can compare this interaction time with the mean time it takes EGR1 to dissociate, right? So this is the consensus binding site, so this time is quite long, and we can see that this is comparable. So this is actually compatible with a passive mechanism. It just waits there until it can move forward. However, so far I just showed you one orientation because EGR1 is an asymmetric protein. It has an asymmetric binding site. So we can now just switch the orientation of the binding site so the protein will bind in the different orientation. So when we do that, we see a much faster passage, right? You can see it here with a much slower interaction time. So at least in this orientation, there must be some type of active mechanism of destabilization of the protein in order to go through. So how does RNA polymerase destabilize the protein? So what we would really like to do is to look at the kinetics of binding and dissociation of the protein as RNA polymerase is approaching, right? But this is a very difficult experiment, so we decided to do something different. So we prepare constructs in which the distance from the binding site to the polymerase is different, and we measure the kinetics of binding and dissociation of the polymerase as a function of the distance to the polymerase. So you can see here, we can unzip this construct. We see the transcription factor here, and then we see the polymerase. So this will be a relatively large distance. If you do now, remember the fluctuation analysis to measure kinetics, you can see that both with and without the polymerase, you see relatively similar kinetics. If you make this distance shorter and shorter, one of the things that you see is that the amplitude of the fluctuations becomes smaller and smaller. And this is because as the polymerase starts to move, it starts to bite into the fluctuating region, right? So this is just a technical effect, right? You have a smaller fluctuating region, but you can still measure the binding and dissociation by looking at this fluctuation suppression. So when you calculate the K-off, we saw something very, very unusual, because what we expected to see is that, if you're far away from the polymerase, you see the same K-off as without the polymerase, right? And this is okay. But then we expected to see that as we become closer and closer, and the footprint, the known footprint for the polymerase from the active site to the end of the footprint, it's about 20 base pairs. So we expected to see that close to 20 base pairs, there will be a destabilization, right? The polymerase, as it approaches something, it has distorts the DNA or something that destabilizes the protein. But then once we get after 20, we shouldn't be able to measure anything, right? Because here we are inside the footprint of the polymerase. But surprisingly, we didn't see anything here. But then we saw many binding events of the transcription factor inside the footprint of the polymerase. That's surprise number one. And surprise number two, the dissociation constant is not like monotonic or constant, but we see this modulation of the dissociation constant as a function of where exactly you bind inside this footprint of the polymerase, which already tells you that maybe this footprint is like an oversimplification, as you all know. Okay, so was the question, what are the structural elements that are actually destabilizing the transcription factor, structural elements in RNA polymerase? So now we use our structural approach of unzipping the DNA on this whole complex to measure the different interactions of the polymerase with the DNA, right? And this is what we see. So what we see is that there are two major regions of interactions. I want to focus on this one, which is about 15 base pairs from the active site. So if you look at the structure of the polymerase, so at 15 base pairs of the binding site, there is this structure called the jaw, which actually holds the DNA downstream together. So it stabilizes the DNA. So in my view, this poses kind of like an interesting question, because if the jaw stabilizes a double-stranded DNA structure, how can it destabilize a protein that wants to bind double-stranded DNA? To me, it sounds kind of like a contradiction. But then we did another experiment in which we measure fluctuations of the DNA, but a little bit away from EGR1 binding site, right? So this is where we always measure, the pink one. So we now measure fluctuations of the yellow one. And we ask the question, what will happen to the fluctuations in the yellow region when EGR1 binds to the pink region, right? So now we are at a distant position of maybe 30 base pairs or 40 base pairs. And what we saw is that we can detect binding of EGR1 to the DNA, even though we are away from that. We are looking at a fluctuating region that it doesn't include the binding site. And binding of EGR1 stabilizes not the closed state, not the double-stranded DNA of double-stranded form of DNA, it stabilizes the open state of the DNA, right? So it looks like binding of EGR1 here stabilizes an open conformation of the DNA here, right? And in my view, if DNA stabilizes the open conformation here, which means that this is the energetically favorable state, if I keep this region closed, then I am disfavoring binding of EGR1. So what we think is happening here is that the jaw region is destabilizing EGR1 by preventing the opening of this region. Okay. And the last thing I want to say, some of you may be thinking, why is it important at all? I mean, maybe it's just nothing, just an anecdote. But if you look at, for example, the LHB gene, and we calculated here the binding energy relative to the consensus site of any possible region on this gene, and you can see that there are many potential binding sites for EGR1 also at the gene body, right? There's nothing special about this gene, EGR1, so we look at 3,000 housekeeping genes, and you can see that there is a very good density of binding sites in all of these genes, and the number of binding sites correlates with the length of the gene, which to me it says that these are there just by chance, just because they are very short sequences. So they're very, very common in any gene. And there is nothing special about EGR1. So there are probably many other binding sites for many other proteins all over the gene bodies, which makes elongation by the polymerase quite a challenge to cope with all these proteins. Okay, that's all I wanted to say today. So I will just mention that every time I said we, it was actually them who did the work: Noam Nago, Sergei Rudnitsky, and Adil Hamis. This is my picture of 4 years ago with Sergei and Adil that were also there, and the weather was a bit different than we saw in the previous picture. Okay, thank you very much. If EGR1 is spread, yeah, EGR1 it's a regulates response to many different stimuli, both hormonal stimulus and environmental things. So it's quite common. Are they? No, we looked. Oh, okay, so that's why we took, for example, this housekeeping genes, because you can also take, for example, I didn't show here, but you can take a subset of genes that are all regulated by EGR1, right? So then the question is kind of like even more accurate. If all these genes are regulated by EGR1, so when the polymerase needs to elongate them, EGR1 is also there, right? And you see the same thing.
Of the protein down, of course, but goes down not via key up, but key on, which was for me. Did you see that?
Yes, absolutely. We see differences in K-on, not only by changes to the core site, we also see difference by changing the flanking sequences. Both of them, yeah. But also, if you look at this and you also play with the concentrations, right? So our model is that the process of binding, it's actually a two-step model. So you have like a diffusion limited step, but then you have some kind of like isomerization tight binding step. And this is the one that must be modulated by the sequence, because other one, it would be really kind of like, how can the protein know that it will reach, you know, so what we have here is nothing special. At least we haven't noticed anything special in what we have here, right? What the yellow region fluctuates between open and close, between double-stranded and single-stranded. And when the protein binds, this is fluctuations, and then the protein binds and it stabilizes the open state, right? Remember, when it binds to the pink region, it stabilizes here, the closed region. So I think that it's some mechanical constraint that if you want to bind at a specific state at the binding site, somehow you deform the DNA in such a way that you favor now the open state. Now we apply a little bit of force, so it opens. There are a couple of works. Sunny Shi many years ago talked about allostery through DNA. And then there's a work by Hagen Hoffman, the Weizmann Institute, that also look at the. Sorry? Double-stranded, yes. What are they? So the nucleosome doesn't bind and rebind. The nucleosome is always there, and the nucleosome kind of like diffuses on the DNA. So it slides on the DNA. Okay, yeah. So what we see is that when you have a canonical H2A nucleosome, so you see movements that are quite fast, but very, very limited, right? So the nucleosome is practically confined to its initial position, and these are probably of the order of seconds. This is what we call fast. When we have H2AZ, then what you see is that you have two types of motion, right? You can really see like a double exponential distribution of the step size. You have two populations of step size, the same ones that you had with the canonical nucleosome, but now you have an additional population which on timescales of a few minutes, right? But with much larger steps. So the system is not experiment sensitive to twist. However, if you look at doesn't matter. If you look at how we do the experiments, so the one figure. Okay. So this whole region here is free to rotate, right? So if there's any twist here, it can be relaxed here. Did I answer your question? No. Because H1? Oh, okay, so first of all, to your second question, right, the tightening is symmetric. Because we can do this experiment and unzip one side or unzip the other side, you see practically the same map. So the tightening is symmetric. So this kind of like indicates an on-dyad conformation, like there was this discrepancy, controversy between on-dyad, off-dyad. But what we see in our experiments is that the chromatosome can actually dynamically switch between being on-dyad and being off-dyad, right? And that's a response to perturbations and other stuff. So to your first question, I think it can be a mechanical effect, right? Because the whole stability of the nucleosome it's in a trade-off between the mechanical energy of binding the DNA and the electrostatic energy of the adhesion between the DNA. So if you now have something that stabilizes the mechanical energy, and you know, just because the persistence length of the DNA it's about 150 base pairs, right? So the DNA is quite not that flexible, right? So if you tighten one side, you are actually stabilizing the mechanical energy, right? And then you shift everything to a more stable particle. This can absolutely depend on DNA sequence. So we have done quite a lot of work using different sequences. We don't like 601 that much. In general, we try to avoid it. Here we had to use the 601 because we really needed, in order to do this type of thing, right? That you want to probe many times the same region without breaking the nucleosome. It's very important that the nucleosome doesn't move. And we know that in other sequences that the nucleosome moves. So here we use 601. Thank you. Yeah, actually, I think I would say it's unlikely because the way that we do most of our experiments, even if not all of them, is that, well, I don't know if that will answer your question, but in generally, we expose the nucleosome to H1 in the optical tweezers, and then we move to a region where there's no H1. And then in many of our experiments, we kind of like see how long it takes until you lose it, right? And it's either you have it or lose it. So we don't see like an intermediate, but I can't be like 100%. Oh, yeah. Because we also did an experiment in which, let's say that you see the suppression of the fluctuations for something that happened over there. So we immediately unzip the DNA. So we go and check, is EGR1 bound there? And in 100% of the cases, we find the EGR1 there. Dynamic of the opening and closing of the DNA. Well, it's not necessarily identical because this dynamics depends a little bit on the sequence of the DNA, on the length of the fluctuating segments, right? They're all very, very fast, right? So our method works with all of them, but it's not exactly the same.